Assalamualaikum and hi. This is a bite-sized learning nuggets for blood transfusion. I am Dr. Farida Idris from UPM, and our topic for today is the biochemistry of ABH antigens. So before I discuss about the biochemistry, it is important for you to know about the encoding genes in the formation of ABH antigens. There are three separate loci of genes that are involved in the formation of these antigens. The first one is ABO gene, which present in the chromosome 1. The second one will be H gene, or is also called FUT1 gene, and the third one is secretor gene, or also called FUT2 gene, and both these are present on chromosome 19. This gene encodes for transferase enzyme that which adds a specific sugar to the oligosaccharide precursor chain. So there are two basic types of oligosaccharide precursor chain in which ABH antigens are built. Type 1 and type 2. The difference between these two types are the linkage between the galactose and N-acetyl glucosamine in the precursor chain. The type 1 chain mainly present as a form of glycoprotein and in present mainly in secretion such as saliva and body fluid. The second chain or type 2 chain is mainly in the form of glycolipid and present on the red cell membrane. Okay, if a person has H gene, and most of us have H genes, so this gene will encode a specific transferase enzyme, and this will result in the addition of the L-fucose to be added to the precursor chain, either it is type 2 or type 1. So this will produce H antigens, or is also called H substance. H antigens or H substance are the precursors of the A and B antigens. Without H antigen, A or B antigens cannot be formed. So when an individual has A gene, this A gene will encode for a specific transferase enzyme, and this enzyme will make the N-acetyl galactosamine to be added to the H substance or to be added to the H antigens. So this will convert the H antigen to A antigen. For B individual, meaning that for B individual with B genes, the gene will encode a specific transferase enzyme, and this enzyme will lead to the addition of D-galactose to be added to the H substance. So this H antigen will be converted to B antigen. So if there is no A or B gene in an individual, so there will be no A or B gene will be formed, and this person will be called O blood group. If both A and B genes are present, some of the H antigens will be converted to A and some will be converted to B antigens. So both A and B antigens will be present on the red cell surface membrane. It is important to remember when more A or B antigens are formed, there will be less H antigens present on the red cell's membrane. So the blood group O will have many H antigens on the red cell's membrane, but blood group AB will have the least H antigen on their red cell's membrane. So as a summary, there are three genes, H, ABO, and secretor genes that are important in the formation of ABH antigens. H antigens or H substance are the precursor to the formation of A and B antigens. blood group O red cells will not have A and B antigens on the red cells, but will have a lot of H antigen on the red cells. So that's about it. Thank you. We'll see in another video.